right here. We got purple flowers, we got red flowers. That's controlled by one gene. We've got long versus round pollen, controlled by a second gene. So how many different combinations of those traits can you make? Those 4. You can have purple with long, you can have purple with round, you can have red with long and red with round. So the question is asking you which of those 4 phenotypes are the parental types. So how do you figure, and which are the recombinant types? So how do you figure out which are the parental types? So I've told you something about the parental individuals. What are their phenotypes? So what phenotype is big P over big P, little l over little l? Purple round. Okay. So that's a parental type. And then little p over little p, big L over big L is recessive. P is red, and a big L is dominant, long. So B and C represent the 2 parental phenotypes. So that means that purple, long, and red and round are the recombinant haplotypes, or phenotypes. These are the phenotypes not seen in the parents. So it should have been A and D. Results from class. Most of you, half of you, a little bit less, picked A. A few are also picked D. So A and D are the 2 recombinant types. Questions or concerns about this point? Clarifications needed? Before I show you the results, does anybody have any question about this question? It's not something we've talked about yet, so I'm just trying to figure out if you know this already, if we need to go through it or not. Let's see. Two-thirds of you think it's true. Smaller than expected number of parental phenotypes means genes are linked. Actually false. We haven't gone over this yet, but this is a preview of things to come. So we'll start talking about that next class. Do you think it's chance? Do you think that ratio is just about 50-50, or is there actually a reason that 2/3 of you chose true? People were just randomly choosing? One head nod. By the end of the class, hopefully when we start talking about the next question, chi-square tests, we might be able to actually ask or answer that question. Was that a random sampling of answers? From a class this size, how many of you, if you're answering randomly, should have chosen true or false? So say there's 80 of you. How many of you should have chosen true if you're choosing at random? 80 students, 2 possible choices. How? 40. Right. So we could do a statistical test if we want to, we don't want to, by the end of class to ask that question. Out of the 80 of you, what was happening? So this is the briefest of recaps about meiosis and independent assortment. Because, what did we talk about last class? What was the big concept? We're talking about genes that are not on separate chromosomes. We're talking about genes that are linked. So how does the fact that 2 genes are present on the same chromosome affect how we approach understanding genetics? And the question that most geneticists that use the chi-square, well, not the chi-square test, but use Mendelian genetics, understanding transmission patterns, genotype frequencies, phenotype frequencies, is to figure out, like I said last class, whether or not 2 genes are linked or not. Are they on the same chromosome or are they different chromosomes? So in this case, you've seen this before. This is what it looks like when you have a dihybrid, heterozygous, big A over little a, big B over little b. And after synthesis, after S phase, the tetrads, the 4 copies of each chromosome, 4 A's, 4 B's, can either line up in one of 2 ways. All of the capital letters could be on one half of the cell, and those segregate into daughter cells so that each haploid gamete has the capital letters, and that means that the other 2 products of meiosis would have the lowercase letters. Or after synthesis, at meiosis 1, the capital letter A's might line up on the same side of the cell with the lowercase b's. And that's how we get to the situation where we have 4 different haplotypes. The big A's with the big B's, little a's with the little b's, big A's with the little b's, and the little a's with the big B's. That's independent assortment. 4 types of gametes from a dihybrid, 4 distinct haplotypes, all 4 possible combinations. And I'm only telling you this again to contrast with something that we didn't summarize last class, was how this differs essentially from what happens when we find A and B are not on separate chromosomes. I'm sorry. Nobody threw something at me. This is the case where A and B are on separate chromosomes. What happens differently when A and B are linked, when they're on the same chromosome? So how does this affect the gametes that we see down at the bottom of this figure? If capital A and capital B are physically connected to each other, which of these 4 gamete haplotypes are we probably not going to see? So under linkage, if the A and the B genes are on the same chromosome, what I tried to write last time, how does this affect what's going to happen? Go through synthesis, you've got big A's with big B's, you've got little a's with little b's, and then you do the first division. So up until this point, I've been trying not to bore you to death, but the big A's and the big B's didn't necessarily have to go together into the same cell at the first division, as we saw in the previous slide, because they're on separate chromosomes, and the chromosomes align randomly. What's got to be true here in terms of capital letters, lowercase letters? If the 2 capital letters are linked, they're on the same piece of DNA, what's going to happen here? Yeah. Those pieces of DNA will move into the daughter cells like this. So that's after the first division. Okay, what's going to happen now? Second division. Of these 4 gametes that we got after unlinked meiosis, that is independent assortment, which of these 4 types are we going to see come out of the case where we do meiosis when the genes are linked? We're only going to get big A, big B's, and little a, little b's. The parental haplotypes. And by definition, by extension, what we don't see when the big A is linked to the big B and the little a is linked to the little b, we're never going to see recombinant types. We're never going to see chromosomes where there's a big A with a little b on the same piece of DNA, or a little a with a big B. So how does linkage affect meiosis? How does linkage affect diversity? Linkage reduces the diversity of gamete haplotypes that are produced. When independent assortment was happening, we got 4 different gamete haplotypes. With linkage, we got 2. That makes it hard for geneticists to combine traits that they want to combine into an organism, because the way we do that is crossing organisms together, sex, the combination of gametes. And if we have cases where genes are linked, they associate together. Sometimes there are cases where we can't take two different traits that are controlled by two very closely linked genes and put them in the same organism because they're linked. Fewer gamete haplotypes produced. The good news is, and some of you have alluded to this last class, you can actually disrupt a linkage. So everything I've said up until this point, if I used any absolute words like never or always, then I apologize, I was lying. There are cases where if big A's and big B's are on the same chromosome, they can actually be physically separated from each other. That's the process of physical recombination. That's the disruption of linkage. And we've already seen these data, crosses from Bateson and Punnett using these flowers that we've been talking about. And when they observed different numbers of organisms with different combinations of traits than they expected, they started assuming, they had to assume, that there was recombination happening. What are the genotypes of these plants that are boxed here, the two middle types? If you have purple flowers and round pollen, what would the genotypes of those plants be? So it could be, purple's big P is dominant, right? So it's going to be big P over something. I'll put a question mark. It could be big P over big P or big P over little p. Either of those would be purple. Then what about round? Okay, so round was recessive. So this has to be big P, little l. Remember, we're talking about linkage. So if the big P is on the same chromosome as little l, wait a second. So you're telling me a genotype that's big P over something and little l over little l makes a purple flowered round pollen plant. How did you get a big P with a little l? Right? The chromosomes came from all of these parents. In fact, the chromosomes in those F2s came from the F1s, where big P is next to big L, and little p is next to little l. So how did you get a chromosome that was big P with a little l? This has to be, if you see any purple flowers with round pollen, you have to have a big P allele. That makes the purple flowers. And you have to have two little l's. That makes round pollen. So how did you get that genotype? Big P over some other sort of P, but two little l's. Means we guarantee you one of the chromosomes had to be big P with a little l. How did we get that? This is what confused Bateson and Punnett when they saw this table. They expected to see some number. They observed actually fewer of those than they expected. They saw very few purple flowered and round pollen plants, but they did see some. Same is true for red flowers and long pollen. This is when recombination, physical recombination, was identified. Somehow, some physical process inside the cell in the F1 generation created a chromosome that had a big P and a little l on the same piece of DNA. We can talk about whether those values are different later. That's chi-square test. So this is where we're heading. We use information about how many observed and how many expected individuals we see to get an idea of whether or not genes are linked or not. That has to do with that question I asked you. When you see an excess of parents or an excess of recombinants, does that mean that the genes are linked or not linked? So by the end of today, maybe by next class, we should have the actual answer to that question. So we geneticists, compare these two columns of information to figure out, are genes independently assorting? That's the expected number. If genes are independently assorting, then the expected and the observed numbers should match. So when we see differences like this, we start to think maybe that means the genes are linked. So that's where we're headed. We're going to talk about physical recombination, meiosis, and then we're going to do a little bit of chi-square analysis. That has to do with cats or dentists or something like that. I have a cool example to share with you. Okay, what's this a picture of? No, you can't say crossing over. How many pieces of DNA are shown here? What is this object? Anybody know? If I diagram this sort of camera lucida style, just drawing what I see, this is a photograph of chromosomes. It looks something like this. So we've got 4 strands of DNA, 4 double helices in this picture. What are they doing? Why are they making these X-shaped structures? They're crossing over. The only thing you really need to remember about this is that each of those is called a chiasma, which is from the Greek letter chi, which looks like an X. So the people that originally saw this under the microscope said, what the hell? What are these chromosomes doing? You look in meiosis, you see chromosomes do this. So this is metaphase of meiosis 1. When all 4 of, this is a tetrad, right? Those are the 4 copies of chromosome 1 after synthesis in a diploid, for example. 2 from dad, 2 from mom. So that's 4 of the same chromosome, and they're crossing over at chiasma. This is when the chromosomes physically break and rearrange parts to make new combinations of alleles like big P's with little L's. And then all of a sudden it made sense. Bateson and Punnett's data, how did we get a chromosome that has a big P with a little l? Well, chromosomes, every meiosis, which they do, physically break and swap with each other bits of DNA. Then it's easy to imagine how bigs and little l's could come together. So after synthesis, if you take this dihybrid, what does this look like after synthesis? You've got a diploid individual. How many chromosomes am I going to draw? 4. And the big P's and the big L's will be here, and little p's with little l's next to each other, because we know that's how the chromosomes pair at meiosis. The 2 copies of the same chromosome are more physically tightly associated with each other. Crossing over. You've seen this before in some sort of biology class at some point. The chromosome physically breaks. I'm just drawing these for example. It doesn't have to happen this way. But in this case, I'm showing 2 of the 4 sister chromatids break in between the genes P and L. So then what has to happen for recombination to occur? How do we get a big P with a little l? How does the cell repair these chromosome breaks? This big P section of the chromosome physically connects to the bottom of the other chromosome. So big P now is physically connected to a little L allele, and the reciprocal happens as well. So the bottom part of the chromosome on the left gets attached to the top part of the third homolog. So now we've got a big P with a little l, and at the same time we also got the reciprocal. We got a little p allele stuck onto a big L allele. So after crossing over, two of the four didn't change. So in this case, the way I drew it, the second and the fourth chromatids didn't participate in breaking an exchange of double helices. Which of the two parents do those look like? Are these parental or are these recombinant types? A big P with a big L, a little p with a little l. And you compare to the parents. These are parental type haplotypes. The combinations we see up here in the parents. Big P's with big L's, little p's with little l's. What did we make by crossing over? We made a big P with a little l and a little p with a big L. Recombinant types. These are the definitions of how you know whether or not your chromosomes are recombinant or parental. And you're going to compare the haplotype, the alleles that are on a single piece of DNA, to the parents of the cross and ask, are they similar or are they different? Is this a new combination of alleles that we hadn't seen before? Any questions or clarifications needed? Turns out, here's the information download from me to you. Recombination events occur randomly across a chromosome, most of the time. So you can't really, in most cases, predict where that breaking of the chromosome will be. It'll be at the end of the chromosome, it'll be in the middle of the chromosome, it'll be 2/3 of the way up. So if you had to guess, if you were a gambling person, what controls the probability? What dictates how likely it is that crossover happens between any two genes that you're interested in studying? Let's draw an example. In which of these two cases is it more likely that there would be a recombination event between genes A and B? Why on the right? Because they're farther apart. So there's a bigger target for recombination to hit. Two genes are farther apart, it's more likely that the chromosomes will happen to break in between them than two genes that are very close together. Which is why it's harder to get recombinant types the closer together two genes are on a chromosome. It's much less likely that by chance, if I was blindfolded, I should do this in class someday. If I was blindfolded and I had some darts, maybe I shouldn't do this in class. And I randomly threw darts here. What's the probability that I happen to land a dart in between the two closely linked genes? It's pretty small. If the two genes are at the opposite ends of the chromosome, what's the probability that a recombination event happens between them? If the two genes are at the absolute opposite ends of the same chromosome, what's the probability that recombination happens between them? So that gets us to the second question. Is there always a crossover? Every time you have this situation, after synthesis phase, before the first division of meiosis, will there always be, in every cell for every tetrad of chromosomes, a recombination event, a crossover? Does anybody remember? You've heard this before, I know. Why is crossing over important? What does it do for the cell? Why do cells do crossing over? Doesn't it seem sort of inherently dangerous to you? Chromosome actually purposefully breaks its chromosomes right before meiosis. Seems like a bad idea to me. So there must be a benefit associated with it, or else evolution wouldn't have evolved our cells to do this. So why does recombination happen?
It benefits from variation.
Okay, so it increases variation. This is exactly the same point that you also made earlier, the counterpoint, that the recombination creates new combinations of alleles, new combinations of gametes, increases diversity, which is often good. At the same time, it exposes our chromosomes to a bunch of potentially bad things, like the cell trying to figure out how to repair its chromosomes. Every tetrad undergoes at least one crossover per meiosis, despite the fact that it's weird, kind of, that our nuclei break our chromosomes at meiosis. This happens. Every tetrad. So chromosome 1, there will be at least one recombination event in humans, every meiosis. Chromosome 2, same thing. Chromosome 3, same thing. How many crossovers can occur in one tetrad? So when you look at a single one of those, right before meiosis 1, how many breaks get made? It's at least one. That was the second question. Recombination is going to happen for every tetrad. How many times it happens is variable. Depends on the species you're looking at. It depends on a lot of factors. How many crossovers within a tetrad will happen? So you might get something in humans that looks like this, where the first and the third tetrads recombine there, and the second and the fourth recombine there, and the third and the fourth recombine there. So that happens in humans and many other species. For simplicity, for this class, erase that from your minds. We're just going to assume that exactly one crossover happens per meiosis to make things intellectually easy on ourselves. If you want to know more about how recombination really works, that might be our advanced genetics class. So our assumption is going to be 1. I just want you to know that that's an assumption. It's not actually true. It's a simplification. Okay. I want to refresh your memories a little bit. You have genes A and B on 2 separate chromosomes. How many of the gametes that get produced by meiosis would be parental, and what percent would be recombinant? So if the A's and the B's are on separate chromosomes and you do meiosis, then you get 50-50. You get big A's with big B's, you get little A's with little B's, depending on how the A and the B chromosomes align during meiosis 1. And then you also get big A's with little b's and little a's with big B's. So what fraction, this is really important, when genes are on separate chromosomes, what fraction of the gametes will be recombinant? %50. So whenever you see 50% recombinant, that's your clue that the genes that you're looking at are unlinked. They're on separate chromosomes. When you see fewer than 50% recombinants, if you see less than 50%, if you see more parental types, and you see fewer recombinants, then what do we conclude? This is linkage. This is genes are on the same chromosome. So in blue, you see more parental genotypes or haplotypes and fewer recombinant haplotypes. That suggests the genes are on the same chromosome. Then all we have to do is figure out how we actually identify the haplotypes of gametes. That's relatively straightforward. It involves some crossing, but we can handle that. So I'm going to skip this fancy looking thing. Oh, I can skip this one too. Perfect. So if genes are linked, they won't make 9:3 to 3:1 ratios of phenotypes. That's what independent assortment predicts. We talked about that last class, how to go from the 1:2 to 1:2 to 4:2 to 1:2 to 1 ratio, etc. 9:3 to 3:1 phenotype ratio. The farther apart two genes are, the more likely it is that recombination separates them during meiosis. So together with those 2 points, if you can figure out how many recombination events happen between 2 genes, you can use that to measure how far apart the genes are. If there are lots of recombination events that you see during meiosis, then the genes must be farther apart. If you see very few recombination events happening between 2 genes, you infer, as we did earlier, that they're really close together. Less likely by chance that recombination happened to occur in between 2 genes that are really close together on the same chromosome. So that's where we're headed. Next class, although we're not to next class yet, we'll talk about using recombination events to map genes, recombination mapping, genetic mapping. But before we do that, we have to talk a little bit about the chi-square test. I don't do my best. I wish I did. I try, but I don't consciously do my best to help you understand the relationship between the material we're covering in class and how it might apply to you in the real world. So, if those of you who are really good at probability and statistics might win at poker. That's the best I can offer you for this. Yeah, it's not much incentive, I know. How about this then? I was thinking about last night, how could I try to make this relevant, and how, even if you're not interested in being a geneticist or using genetics, how can I try to convince you that probability and statistics is actually useful to understand? So for those of you that might be skeptical, I went to Google and I typed in 4 out of in the search bar, and these are the most common results. 4 out of 5 stars, 4 out of 5 dentists. So apparently a lot of people type 4 out of 5 dentists into the Google search. So what is it that 4 out of 5 dentists are doing? Well, apparently, according to the results, they're recommending Trident gum, or they're recommending a particular type of toothpaste. You've seen the commercials, probably, right? 4 out of 5 dentists recommend you rinse before you floss, whatever. What does that mean? It's supposed to convince us to do something, right? These are advertisers trying to make us buy their product. Me personally, I'm just going to share my half-a-minute story. For me personally, the reason to understand probability and statistics, the reason that genetics class is really useful in the real world to everybody is to try to make you think critically when you hear numbers like 4 out of 5 dentists tell you to buy their toothpaste that they're hawking. Right? Question everything, especially when it has to do with numbers or statistics or probabilities. What would you like to know about the 4 out of 5 dentists? Do you believe those ads? Did you go buy their dental floss? You saw that. know what Russian roulette is, Google it, but, right? What does it mean? What do you imagine it means when somebody does a study and they say 4 out of 5 dentists recommended it? What do you imagine in your mind some policy institute did? Somebody called 10,000 dentists on the phone and asked them what they prefer? What could this mean and still be true? 4 out of 5 dentists recommend our chewing gum. Maybe somebody just asked 5 dentists, and 4 of them said, yeah, I recommend your gum. Should that convince you to buy the chewing gum? What would you like to think happened? Somebody surveyed 100,000 dentists, and 4 out of 5 of them said, yeah, your gum's good, right? The bigger numbers, maybe, the more people that have been contacted, discuss regarding this topic, that maybe that should sway us. But if it really is just 4 out of 5, they don't tell us. The assumption is, 4 out of 5, it's a ratio. 80% of dentists, this is the way I interpret it anyway, this is the way the advertisers want you to think about this. 80% of dentists, if I asked them, would tell me to buy this gum. But these advertisers wouldn't be lying if they had only asked 5 dentists and it happened that 4 of them had said, yeah, use our gum. This is why probability in the chi-square test is critical. Right? It lets us think about, okay, how significant are the data? Did somebody really just ask 5 dentists, or did they ask 10,000? At what point should we actually believe what we're hearing? So this makes it personal for me, which doesn't mean it makes it personal for you, but I've got 2 daughters, which I think I've mentioned before. Therefore, do you have any evidence that I have a Y chromosome? If I have a son, then I have genetic proof that I have a Y chromosome. Because it's my chromosomes that determine the sex of my offspring. If I give them an X, they become females. If I give them a Y chromosome, they become males. So could you use this as proof that I don't have a Y chromosome? So with just a small sample size, what does this mean? How many sons should I have had? If I have two offspring, what would you predict? How many sons and daughters should I have had? What was that? One of each. Right, I've got 50% X chromosomes, 50% Y chromosomes. I should have an equal ratio of sons and daughters. That's how our population and our species doesn't go extinct, by the way. It's kind of important to have an equal number of males and females on the planet, because if you kind of start going towards one direction, then pretty soon you run out of people to mate with. Okay, so the expectation is that I would have at this point 1 son and 1 daughter. So is it significant? Does it mean anything that I haven't had any sons yet? Or is it just chance? It's just bad luck that by chance I've only had daughters. Don't tell them I said that. So if I've got 2 females that are observed, and you just told me you expect to see 1 female and 1 son, the important thing in the chi-square test is to make sure that the number of observations and the number of expectations are the same. So your expected 1 plus 1 should equal 2, the same as how many total observations have been made. So here's the quick recap on the chi-square test. The formula, which you might remember, is you take the observed value for each of these lines. You take the observed value and the expected value, you square them, and you divide by the expected value. Is that reasonably familiar, even those of you that did this 5+ years ago? So what you have to remember is to do this for every different category. In this case, we have 2 different categories. We have a row for the XXs, the females, and we have a row for the XYs, the males. So if we filled this out, this would be observed females, 2 minus 1 expected, square that, divide it by the expected value again, that's 1. So that's the top line. And then we'd do the same thing, we'd add that to the next observed minus expected squared over expected for sons. So 0 observed minus 1 expected squared divided by 1. So when you do that relatively simple math, the result that you get is the chi-squared test statistic. That's its formal name. You get a number. What does it equal? 2 minus 1 squared is 1 squared, which is 1, divided by 1, which is 1, plus negative 1 squared is 1, divided by 1 is 1. So the chi-square value here is 2 for my specific case. How do we turn this into a probability value? This is the chi-square value. That number 2 shows up somewhere in this part of the table. What else we need to know is the number of degrees of freedom, which may give you flashbacks or nightmares or something horrible. Degrees of freedom is easy. What is it? If you remember, if you were confident enough to tell me what a p-value means, you probably are confident enough to tell me what how to calculate degrees of freedom. It's the number of, it's the number of categories, essentially, minus 1. So we've got 2 categories. It's always minus 1. That's the degrees of freedom. In this case, 2 minus 1. We're looking at 2 different phenotypes, boy versus girl. So 2 minus 1 gives you the degrees of freedom. So that focuses our attention on this line of the table where df, degrees of freedom, is 1. So the way you turn with the chi-square value, you calculate using the chi-square formula, and the number of degrees of freedom, you read from left to right across the table until you hit the chi-square value, which in this case was 2. So the number 2 falls there somewhere in between 0.016 and 2.7. So that's the range in which our chi-square value of 2 falls. So the last thing you do is you look up from there to the header of the columns in this format of the chi-square table. And these numbers that are the subscripts here, 0.9 and 0.1, those are p-values, probability values. So in this case, a chi-square value of 2 has a p-value that's between 0.9 and 0.1. That's the answer. So chi-square test. You get a bunch of data, you have observations and expectations, you perform the chi-square calculation. Then you calculate the number of degrees of freedom, you find your chi-square value in the table, and then you look in the header of the columns to find the p-value. So what does a p-value mean if it's between 0.9 and 0.1? So when a p-value is between 0.9 and 0.1, is it significant or is it not? So I see 1 thumbs down. Scientists, and this is just something you have to remember, scientists have decided that the p-value has to be less than 0.05, smaller than 0.05, the p-value, for there to be a statistically significant what? Between the observed and the expected values. So fill in the blank. This is the part that's hardest to remember. 0.05, that's easy. Is it a statistically significant difference or a statistically significant similarity? That is, what was it? Difference. So whenever you find a chi-square value that has a p-value of less than 0.05, that means the observed values are not equal to the expected values. There's a significant difference between them. So in my case, my p-value was much bigger than 0.05, which means the observed and expected values of my offspring's sexes are not different. So having 0 sons at this point means nothing about the absence of my Y chromosome. So I know we're now right out of time, but I want to leave you with these last 2 questions to think about for next class. Here's a case of a family that had 12 boys in a row. They have 12 kids and they're all males. So of course I'm going to ask you for next class to calculate, is that significantly different? What would you expect a couple with 12 kids to have in terms of ratios of sexes? And is that significantly different from having 12 sons and 0 daughters? Okay. By the way, the 13th kid that they had was also a boy. So now it's 13 to 0. Okay. So come back to class next time, we'll continue discussing this.